In this session, we will learn about immunofluorescence. So, practically speaking, immunofluorescence is a staining or visualization technique. But this is not a common staining technique. There are some specialities which make it very unique technique. The first unique feature is that instead of staining whole cell, only specific parts or molecule are stained or labeled. This molecule can be present on the surface of that particular cell, or even they are present inside the cell. So what we can achieve by staining only specific parts or molecule of a particular cell? One thing we can check whether that particular molecule is present or not inside the cell. So we can check its presence. And other thing, if that particular molecule is present in the cell, what is its exact location, or we can say how it is distributed throughout the cell. Second unique feature of this particular technique is stains used. In this staining technique, special stains which are known as fluorescent stain or fluorochrome are used. These fluorochromes are connected to monoclonal antibodies, and such antibodies which are connected to fluorochrome are known as fluorescent antibodies. These two parts, that is the fluorochrome and the monoclonal antibody, have two different functions. So as I told, in this technique only specific part or molecules are stained. So these antibodies are used for that particular purpose. These antibodies specifically bind to the desired part only. So if we want to stain nucleus of a cell only, so we can use anti-nuclear antibody, that is the antibody which binds to nucleus only. The fluorochrome, on the other hand, it emits light of a specific color when excited with another light of a specific wavelength, and this mechanism in which that fluorochrome emit the colored light is known as fluorescence. So in this way, only desired part of that particular cell can be visualized as color emitting area. So in this photograph we can see nucleus of a cell emitting blue color. Different fluorochrome which are connected to different monoclonal antibodies, they can be used at the same time to visualize two or more part of the cell. So as we can see in this photograph, many part of the cell are stained at the same time. There are two types of immunofluorescence: direct immunofluorescence and indirect immunofluorescence. In case of direct immunofluorescence, fluorescent antibodies directly bind to the target molecule, that is target antigen, like this. But in case of indirect immunofluorescence, first a primary antibody, that is which is not connected to the fluorochrome, it is added and it binds to the target antigen like this. And after its binding, then a secondary fluorescent antibodies against the primary antibody, they are allowed to bind them. So in this case, the fluorescent antibodies, they binds to the target molecule, the target antigen, indirectly, that is through the primary antibody, and that's why this technique is called as indirect method. So this indirect method, it is more sensitive than the direct one. Why? Because as you can see, in case of direct method, only one antibody, only one fluorescent antibody is bound to per target molecule. But in case of indirect method, two or more antibodies are bound to the primary antibody which is bound to the target. So that's why in this case light emitted will be more, and that's why image will be very clear, and that's why this method is more sensitive. Now we will see the actual protocol. The first step is the sample preparation. In sample preparation, the target tissues or target cells, they are embedded in paraffin wax like this, and after that very thin slices are prepared using an instrument which is known as a microtome. So here you can see the microtome making very thin slices of that particular tissue. After this, selected thin slice of that particular tissue is transferred on clean glass slide. And after transferring that slice, the wax is removed with the help of de-waxing agent. And in this way, the slide is ready for staining. Suppose we have to check the distribution of a protein called as microtubule by direct immunofluorescence. For that, first we will add fluorescent anti-microtubule antibodies on that thin slice of the cell and incubate for some time. During incubation period, these antibodies will bind to microtubule present in the cell like this, and many antibodies can remain unbound. These unbound antibodies can be removed by washing the slide with the help of buffer. After this, the slide is observed under fluorescence microscope. Fluorescence microscope will bombard the slide with a light of a specific wavelength, and this light will excite the fluorochrome to emit a colored light from microtubule, and therefore our microtubule, the target, can be seen as color emitting area like this. So in this way, different parts of cell can be visualized using direct and indirect immunofluorescence. These are some examples of fluorochrome, the wavelength required to excite them, and the color emitted by them. Thank you very much for watching this video.